Hello, so my name is Josef Penninger. I'm the head of the largest life science institute in Canada, in Vancouver. I'm actually a trained MD and a genetic engineer, so we did some of the first mutant animals, which led to cancer immunotherapies. We discovered the master gene for bone loss, which became a $5 billion annual drug. And I also co-discovered ACE2, so which of course turns out to be the gateway for the SARS-CoV-2 virus to infect us. And we had the first paper showing it's the essential receptor for the virus. So along the way, doing genetic engineering since more or less 30 years, we were interested in how we could adapt cells live to extreme conditions. And of course, how could we actually make cells live on Mars? So how could we actually accomplish this? And of course, this environment, extraterrestrial and also some terrestrial environments, are very different and needs adaptation of life. There's reduced oxygen on Mars. There's some, but much reduced. There's a sort of gravity affects cell adhesion, how multicellular organisms actually adhere to each other, how they talk to each other, how cells divide. There's 150 times more radiation. So how could we accomplish to adapt cells to such environments? Our life as we know it has shown this already. It underwent 4 billion years of adaptation. So from a fiery bowl to a world which was full of ice. So it took evolution more or less 4 billion years to evolve complex genomes and organisms as we know them now, which are living on Earth, leading, at least in humans, to a diploid, so a duplicated human genome of around 6.4 gigabases. So the question was really this, which we asked some years ago. It was completely crazy. Could we actually develop systems and technologies which speed up millions and billion years of evolution in one lazy afternoon? Could we basically play the game of evolution in a day? And to do this, we first had to trick biology, and we did this, we and actually other laboratories. The trick was actually to create cells which should not live on this planet, and actually normally don't, to halve the genome of stem cells. So normally mammals and basically all other living organisms have 2 sets of chromosomes or diploids. This protects us against mutations. Because for us in genetics, that's a real problem because we want to set mutations to understand life. But if we have a singular genome, a haploid genome, then we can readily set mutations. Also, we can combine this, having stem cells, with tissue engineering. So for instance, some years ago, my lab developed the first perfect human blood vessels, which we published in Nature. We were involved in creating the first human brains from stem cells. So basically, this was the major thing to break one of the paradigms of biology. And now the idea is this. We can take the cells, have a halved genome, and we can combine this actually not with advanced genome engineering, because we actually do very simple genome engineering, which was developed 100 years ago. The reason is this actually goes beyond what people are doing now, because it allows us to screen the entire genome in an unbiased way at the single nucleotide resolution. So in an unprecedented resolution. So we basically expose then the cells to altered metabolism, low oxygen, as it is on Mars, change microgravity, expose them to radiation, and basically define the mutation in an unbiased way without knowing anything, and find cells which can live under extreme extraterrestrial and also extreme conditions on Earth. So this is not just utopia. Actually, we have shown this works. For instance, using this technology, we can do basically structure by genetics. Many drugs out there, we actually don't know how they work. So using our technology, we can actually pinpoint the exact amino acid and nucleotide responsible for drug action, which really opens up a new universe of drug development and understanding how drugs work, of course, combined with what people are doing in the AI and the predictions of structures. We also using the system to figure out the inner workings of viruses infecting our cells, because they actually hook onto ancient machineries ancient evolutionary machineries. For instance, the SARS-CoV-2 virus to replicate in our cells. And using our technology, we can actually figure this out and find the weak spot within our cells the virus needs. So to my knowledge, there's actually no other technology we could do this. Because for instance, if you do CRISPR engineering of genomes, you will actually kill particular proteins and we will never find them. So the trick for us is to mutate the entire genome at a single amino acid resolution. And by doing so, we can pinpoint the exact site, an unprecedented, unbiased resolution. So as Gustav Klimt already painted, through fundamental knowledge and understanding. So we, the scientific community developed amazing technologies. And I think some of the technologies I showed you can allow us to understand the tree of life, and of course might allow us to actively change the tree of life. And in the world we live now with changed climates, which requires adaptation, and I think it's high time to actually do this. And of course, one day this might really allow us to adapt ourselves to live. It's the grand challenge we have to live on Mars. And with this, thanks for listening to my little idea.